Yes, sir. Biomolecule.
Okay,
Sir,
we were
but
doing biomolecules.
sir, but today class we can take only for 1 hour.
Okay, you have to be somewhere.
No, sir. I have Learnivio test. That's why.
Okay, then we should start. So just tell me, like, have we done this cyclic part? I think we need to discuss how the mechanism works, right?
Yes, sir.
Okay, so let us start then. First of all, I would request you to please turn on your camera. Right, let's start. So, in the theory, we understand this straight chain is going to get converted into a ring. How? The carbon on the fifth number. If I start the counting from here, this carbon on the fifth number will be attacked from the first one. How it is going to attack, that we need to discuss. Right. So let us take this to a new page. See, we have a glucose structure. It looks like this. Okay, at the top we have CHO, then OH, H, OH, OH, and then this. Is this correct or not?
Yes, sir.
Okay. So try to think. We need to connect somehow. Okay, one, two, three, four. This fifth carbon with the first one. So what we are going to do? We are going to rotate this straight chain. This chain is flexible, so we can rotate it like this. So if we do that, what we are going to see is this. Okay, let me check. Uh, it's me. Right. So, yes. So we have this variable bond OH. I am trying to rotate it like this. This CH2OH at the top. Suppose first of all you need to do this. You have your ring. You are doing what? You are rotating it like this. Okay.
Yes, sir.
Now this CH2 group is here. On the next we have this COH. Then we have carbon. Then we have again carbon. 1, 2, 3, 4. Then 5. Then 6. Here we have double bond and then H. Try to understand. If the OH group is at the right side, if the OH group is at the right side, where we are going to put it? At the upward area, right?
Yes, sir.
Here, if I count it, this one was the 6 number carbon. 6, 5, 4, 3, 2, and 1. On the fifth carbon, the OH is at the right side. So I placed it above and H below. On the fourth one, again, OH is here and H is here. On the next one, OH is at the downside and H is here. And for the next one, again, OH here and H here. Now what we are going to do? We are going to rotate this. Suppose this CH2 group will go there. This OH will come here like this. We are rotating this. Why we are rotating this, you will understand. So is this clear what we are doing? We are replacing CH2 with OH simultaneously H. Like we are rotating this thing. So what will happen? Now it will look like this. Now our OH is at here. CH2 OH will go this way. C, 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 and OH, H, H, OH, OH, H. Why we did this? First of all, we need the contribution of fifth number of carbon, right? So from here, what we are going to do, this OH has electron pair to donate, right? And if I look at this side, due to the electronegative oxygen, this carbon is having a partial positive charge, and this one will have partial negative charge because oxygen is more electronegative than our carbon. So this oxygen having lone pair will attack here. Here we are using some sort of organic chemistry, attacking type of stuff, nucleophilic reaction, right? So this will attack here, due to which this bond will go on the oxygen because carbon cannot form more than 4 bonds. It will have to break one. So then what will happen? It will look like this. O negative. Here will be a bond, right? This will be CH2 OH. We will assign these respective groups like this. Now, what we are observing? We are observing a bond. This new bond, right? Because the oxygen donated its own lone pair, it is having a positive charge on it, right? Because it donated
Yes,
some
sir.
electrons from itself, it has positive charge on it. And this oxygen has negative charge. This electronegative element, which is oxygen, will not bear this positive charge because oxygen's tendency is to gain electron because it is electronegative. If you are taking an electron from oxygen itself, then it will be very unstable. So it will try to remove this positive charge and get neutralized. So it will do what? It will lose these, it will lose this hydrogen and take these two electron pairs to neutralize itself, to remove this positive charge. And this O negative will take this oxygen. This H positive will be removed and it will be uh, taken by this O negative, and these electrons will be transferred to the O positive. So if I do this, then what I'm going to see? Now it will look like this. Okay, and
Sir.
this had its own H as well. H. Right? So it will have its H like here. Now what we need to understand, this is our structure of cyclic glucose. Let us check is this correct or not. This is let me see, this is wrong. We did something wrong. I did something wrong. Check. This is our this one. And this is the lower end. Okay. One correction. The oxygen which is at the right side will be placed on the lower end. It will look like this. The oxygen which is on the right side will be placed below. So we need to correct all of them. Now this is correct. Okay. So we observe there are two forms of cyclic structure, right? One is alpha, one is beta. So how we will decide the alpha form is forming or beta form is forming? Suppose if the attack of this oxygen, if the attack of this oxygen is from the backside, if the attack of this oxygen is from the backside, and if the attack is on the front side, if the attack is from the front side from here, let me change my color. If the attack is from the front side, this one, then the product that will be formed is our this beta form. If the attack is from the first side, then the product will be formed is beta, because then the OH group will be at the top side. If the attack is from the back side, then the product formed will be this alpha one, because now the OH group is at the front side. So here what we did, suppose here, let us do the attack from the back side. If the attack is from the back side, this OH group will be formed lower end, and this one is alpha form. If the OH group is at the top side, then it will be beta form, right?
Yes, sir.
So what was the first step? First we rotate it to 180 degree, right? Then we like rot bend it a little bit. Then we did the rotation. This carbon, this hydrogen, and this OH group will be rotated. Why this is rotated? Because we need to make the bond through this carbon, and the bond is done through this oxygen. So this oxygen is needed to be here. So we need to do the rotation stuff, and we will get this orientation. Once we get this, we will simply attack. This will be transferred here. This will get positive charge. Then it will gain this H positive, and we will get our product. The thing that you need to understand is the attack. If the attack is from the back end side, the product will be alpha. If the product is from the front end side, the product will be beta. Or you can simply understand, if the oxygen, this OH group is at the lower end, then it will be alpha. If this OH group is at the top side, this will be beta. Okay.
Sir.
And the carbon on which this OH group is, it is anomeric carbon. Okay. One last thing. If the OH is at the right side, it will be placed at the below part, below the plane, right? I made the mistake. You should not do the same. Now we talked about glucose. So the next thing is fructose. Fructose and glucose have same molecular formula. The position is different. In glucose, we have aldehydic part, but in fructose, we have ketoic part, right? They both are hexa compounds, both have 6 carbon chain. So let us understand what is fructose. Fructose is a ketohexose. Everything is same, but the aldehydic group is replaced by ketone group, right? It is obtained along glucose by hydrolysis of disaccharide of sucrose. If we are boiling sucrose in acidic medium, we will get 2 units, one glucose and one fructose. We have already discussed this, right? In the preparation method part.
Yes, sir.
If we have sucrose, if we are heating it, if we are boiling it in acidic medium, acidic water, then we will get one fructose and one glucose. Now let us talk about structure of fructose. Fructose has same molecular formula, and on the basis of its reaction, it was found it contains a ketoic group, and the ketonic group is at the carbon number 2, right? It belongs to the D series. What do we mean by capital D? It is a relative configuration, right? Capital D is relative configuration, and plus minus is, or we can say small d, which is dextro and retro, are absolute configuration. Capital D is relative, right? It is related to our glyceraldehyde isomer. If the OH group is at the right side, we will call it D. If the OH group is the left side to the last carbon, last chiral carbon, then it will be called L.
Yes, sir.
And it is levo rotatory compound. How do we know it is levo rotatory? Because it is rotating the plane polarized to the left side, and it is a purely experimental phenomenon. Its name is written as D. D means it is related to glyceraldehydes D conformer, and minus it shows that it is rotating the light to the left direction. Minus is showing it is rotating the light to the left direction, levo. And fructose, it is written like this. Its open chain structure is shown as this. This is the open chain structure of fructose. Here, what is different? On the second number carbon, we have this ketonic part.
Yes.
Apart from it, everything is same. If we see these both structures simultaneously, what we will observe. Right. So what is different? On the second carbon, we have this ketonic part, and the rest is same.
Yes, sir.
Right. So here again, this fructose structure will exist in chain form as well as in ring form. So we will talk about, we don't have to go through how we came to the conclusion that it also exists in ring part. We just need to understand the structure. Right. From here, they will only ask about the structure. They will not ask about the methods, how we came to this conclusion. So similar to our glucose, it also exists in two cyclic forms, which are obtained by the addition of OH at the carbon number 5 again. The carbon number 5's OH group will be attacking on the carbon number 2. This carbon number 5's OH will attack on the carbon number 2's carbon, basically, right? This carbonyl part. The ring thus formed is 5-member ring and is named as furanose. This is how our furan look like. With the reference of this furan, we name this furanose. Right. Furan is a 5-member ring cyclic compound with 1 oxygen and 4 carbon. So let us understand how these structures look like. Here, again, we will rotate it by 180. We don't have to go to the mechanism because they won't ask you the mechanism about fructose. So let us understand it critically. Just we rotated it, then we do the conversion. Then this OH will attack here, right? If this OH is attacking at this carbon, it will go like this. Similar method, the one previously we did. Here, what is different? Instead of getting 6-member ring, we are getting 5-member ring. Why? Because this how many atoms are participating in this ring formation? This oxygen and this carbon, and this one oxygen atom. So there are total 5 atoms which are participating in the ring formation. So it is going to be a 5-member ring. It will look like this. These type of structures are called Howard projection. If the OH group is at the below side, then we will call it alpha. If the OH group is at the top side, we will call it beta, similar to our glucose. The full name is going to be alpha D, which is relative configuration, minus it means levo, and the name is fructofuranose. Here, beta fructofuranose. Right. Please draw this structure right now. I will show
Yes, sir.
you how we are going to convert this into this. Let us talk about the mechanism as well. So you can understand about that.
Sir, but it is the same, right?
The
Uh,
mechanism is
when
same, but let us do the first two steps.
no, sir, when it is alpha, then uh, O will bond from the back side, and when it is beta, it will bond from the front side. Same, right?
Okay, good, good, good. If the
Okay,
OH
sir.
group is at the bottom side, it will be alpha. If the OH group is at the top side, it will be alpha. Same
Okay, sir.
like before. Just try to draw it down right now. Then I will give you a brief about the mechanism as well. You should also remember, if we are writing the glucose or fructose in this kind of manner, then we will call them Harvard structure or Harvard projection, right? They might ask
Yes,
you
sir.
which type of structure are these, so you have to check the Harvard thing. So first of all,
Yes, sir.
okay, what is the first step? Here, we need to rotate it like 180 degree, right?
Yes, sir.
After rotating 180 degree, we have to bend it like this. We have rotated it by 180 degree. We have our CH2OH here, this OH like this, or let me correct, it will be like this because this is at the right side. If this is right side, it will be carbon, carbon, 1, 2, 3, 4, then this fifth, and this CH2OH. Right? Is it clear how we draw this?
Yes, sir.
Now, if we need to do the counting, then we will go like 6, 5, 4, 3, 2, and 1. Right? So, which carbon's OH group is going to attack? This one, and it is going to attack at this position, right? So,
Yes, sir.
what we need to do? We need to rotate it. We need this OH group here. So, we'll do the rotation like this. Now we have this CH2OH group here, this OH here, and H here, right? Now what will happen? This oxygen will attack with its lone pair to this position, and this will shift here. Now, it is going to form 5-member ring. Let us draw a 5-member ring. Right? If oxygen has donated its own electron, it will bear a positive charge. And as it has been transferred, like this single bond is transferred to the oxygen, then it will have negative charge. This will abstract this proton, and these electron will be given to oxygen because the positive charge is very unstable on oxygen, right? So if we do this, right?
Sir.
So the attack was done through backside. So the product is going to look like alpha or beta. The OH should be at the
Alpha.
below side or alpha, right? So OH should look like this, and CH2OH. If I rotate it a bit, then it will look like nothing but pyranose, right? I do this. So this is how the mechanism works. Same as glucose, right?
Yes, sir.
So the carbohydrates part is complete. One last thing that I would like to mention is they might ask you the type of bonding. In carbohydrate, the bonding, the linkage that we use is glycosidic linkage. if we are connecting two monosaccharides. If we are connecting two monosaccharides, the bond that will be formed between them is going to be glycosidic bond. They might ask you in one number, which type of linkage is in carbohydrate? So you should answer glycosidic linkage, right? Carbohydrate is complete. So now let us jump to this proteins. So what are proteins? Proteins are basically macromolecules, polymers formed by amino acids. If you have several number of amino acids and you are connecting them with each other, the end product that you are going to get is protein. Protein is macromolecule of amino acids. If you have several amino acids, you connect them with each other, you will get a protein. A total 20 different type of amino acids are attached with each other in long chain to form a protein. We need 20 types of different proteins to form a, 20 type of different amino acids to form a protein. If we are changing the sequence, we will get a different protein. I will show you how we are going to change the sequence. So let us talk about protein first. Proteins are the most abundant biomolecules in the living system. They are chief sources of protein. Like the chief sources of protein is milk, cheese, pulses, peanuts, etc. Right? Protein you can get it from here. They occur in every part of body and form fundamental basis of structure and function of life. Right? These are basics. They are required for growth and maintenance of body. Right? The name protein was derived from this proteor, which means primary or prime importance. Right? This word was derived from this. You don't need to learn this. Just a point. Now, this point is very important. All proteins are polymers of alpha amino acids. We have different type of amino acids, but the protein is only formed by the linkage or connection of alpha amino acids, right? If you have beta amino acids or gamma amino acids, you won't be able to form a protein. The protein is formed only by alpha amino acids, right?
Sir.
Triosine was first obtained from cheese. Triosine amino acid was first obtained from cheese. Right? All amino acids have trivial names. Like if we are talking about glycine, glycine is named since it was sweet, right? And one more thing, glycine is the only optically inactive amino acids. All the other 20 amino acids are optically active. Only glycine is optically inactive, right? One important
Sir.
point, which amino acid is optically inactive? Glycine, right?
Yes, sir.
So let us talk about what are amino acids, right? Amino acids contain NH2 group and carboxylic group. Depending upon the relative position of amino group with respect of carboxylic group, the amino acids can be classified as alpha, beta, gamma. Let us understand what is happening. First of all, we can understand what are amino acids. Amino acids have amino group, NH2 group, as well as carboxylic group. If a compound has this amino and carboxylic group, we will call it amino acids, right? Now the question is, which one is alpha amino acid and which one is beta amino acids? If if this carboxylic group, so try to understand. Just to the next of this carboxylic group, we have this carbon. This is alpha carbon with respect to this carboxylic group, right? This carbon is alpha carbon because it is directly connected to carboxylic group. Or we can say this carbon is alpha to the carboxylic group. And to this alpha carbon, we have this amino group.
Sir.
This is amino group, right?
Yes,
NH2 is
sir.
amine. So if at the alpha position to the carboxylic group, we have amino group, then we will call it amino acid, alpha amino acid. But suppose here to this carboxylic acid, this is the alpha position, right? The
Yes,
directly
sir.
attached carbon to this carboxyl group is this one. But to this we have no amino acid, no amine group, right? The amine group is at beta position, right?
Sir.
So this will be called beta amino acid. So the question is, are beta amino acids capable of forming proteins? No, only this alpha amino acids are capable of forming proteins, right?
Oh,
Suppose,
yes, sir.
suppose we have NH3 here, NH2 here. Now this is which type of amino acid? After beta, what comes? Gamma.
Gamma. Gamma
So it will
amino acid.
be gamma amino acid, right? Is this clear?
Sir. Yes, sir.
Now, classification of amino acids. Amino acids can be classified on the basis of their occurrence, like whether they are essential or non-essential. Whether they are able to form in our body on their own, or do we need to take them externally. So we will talk about them. So first type is non-essential amino acids. Non-essential amino acids are these: glycine, alanine, glutamine, proline, and tyrosine. You need to learn them because they might ask you which are the non-essential amino acids. So you need to know these are the non-essential amino acids. So what are non-essential amino acids? First of all, those which cannot be synthesized in the body and must be obtained through diet. These amino acids cannot be formed on their own inside our body. We need to take them externally, right? These are non-essential amino acids. I think
Which cannot be synthesized are essential.
I think I said the wrong thing. The non-essential amino acids are those which do not need to be taken from external means. They can be synthesized on their own in our body, right?
Yes, sir.
Non-essential. That's why they are non-essential. Example are these. And the amino acids which are needed to be taken from external sources are called essential amino acids. Essential amino acids are these. Okay. Is it clear? What are essential? Essential
Sir.
are needed to be taken from external sources, and non-essentials are created on their own in our body.
Sir.
Amino acid is basically a compound which have a amine group and carboxylic group, right? If we have equal number of amino and carboxylic group, then it will make a neutral amino acids. Suppose, let me complete this first. If the amino group is more than carboxylic group, then it will make it basic. And if carboxylic group is one, then it will make it acid. Let us understand what this is. Suppose the primary requirement for a amino acid is it must have a carboxylic group and a amino group, right? This is the primary requirement so that we can call it a amino acid, right? If
Yes, sir.
the carboxylic group and amino group are equal, if one carboxylic group and one amino groups are present, then it will be neutral.
Yes, sir.
But if we have two amino group and one carboxylic group, then it will be basic. Okay.
Sir.
Or if we have two carboxylic group, if we have two carboxylic group like this, it will be acidic. Okay.
Yes,
If
sir.
the amino group and carboxylic group are same in neutral, if amino is more than basic, if the carboxylic is more, then it will be acidic. Amino acids are usually colorless, crystalline solid, water soluble, high melting point, behave like salts rather than simpler amines or carboxylic acids. This behavior is due to the presence of both acidic. So first of all, they are colorless, have crystalline structure, they are water soluble, have high melting points, and behave like salts. They will have salt property because like they have amine group as well as carboxylic group. So they will behave differently. If we are having a simple amine or simple carboxylic acid, the amino acids will behave differently from a simple amine or simple carboxylic acid. Okay.
Yes,
If
sir.
we have a simple amine like this one, or we have a simple carboxylic acid, they will behave differently than these individual entities. Now, this is an important topic, deuterion. What is deuterion? Let us understand. Again, this is mentioned. Which type of amino acid is optically inactive? Only glycine is optically inactive. Apart from it, all the 19, the rest amino acids are optically active.
Yes, sir.
Deuterion. In aqueous solution, the carboxylic group can lose a proton and amino group can accept a proton, giving rise to a dipolar ion known as deuterion. What is deuterion? Deuterion is a dipolar ion. Why it generates? If we are placing our amino acid in a aqueous medium, the OH, the C double bond OH group, the H part will lose, like it can lose hydrogen, and the NH2 part can take that hydrogen. So it will go like this. Suppose we have a aqueous medium, right? This carboxylic group has acidic hydrogen. In the aqueous medium, it can donate its acidic hydrogen to this amine, right? It
Yes,
will
sir.
form a structure like this. This NH2 group has taken this H from the C double bond OH, and this has positive charge, and this has donated its electron, like not electron, this H group, by taking these two electrons, so it has negative charge. This entity has positive as well as negative charge, so we will call it dipolar species, and this is deuterion, right? Ultimately, because the negative and positive charge are equal, this is neutral. It has both the charges, positive as well as negative, but still it is neutral because the amount of positive charge is equal to amount of negative charge, right? Why this happened? First of all, because NH3 is a base, NH2 can act as a base, and carboxylic acid can act as a acid. The acid basically donates H positive, and base basically abstract or accept H positive, right? We talked about Lewis acid and Lewis base in the coordination chemistry. So what is the work of acid? Acid donates H positive and base, sorry, acid donates H positive and base accept H positive. This is all organic part. We should have done organic first. We should. So is this clear?
Yes,
What is
sir.
deuterion?
Yes.
See, I have again mentioned this, except glycine, all other naturally occurring amino acids are optically active. This is a very frequent question from this chapter, so you should know. Most naturally occurring amino acids have L configuration. L amino acids are represented by this and this. Basically, most of the naturally occurring amino acids have L configuration. What do we mean by L? This NH2 part will be at the left hand side. That's it. Now, structure of protein. Protein are the polymer of alpha amino acids. If we have units of alpha amino acids and we are attaching them together, then we will get an entire protein. So the smallest unit of protein is alpha amino acids. They are connected to each other by peptide bond or peptide linkage. In the carbohydrate, we had glycosidic linkage. If we are connecting two carbohydrate. If you are connecting two monosaccharides, then the linkage that will be between both of them monosaccharide will be glycosidic. If we are connecting amino acids, the bondage or linkage they are going to form is peptide linkage. Let us talk about what is peptide linkage. Peptide linkage is an amide formed between these two groups with the removal of H2O molecule. Transmutable when oxidation units amino acids. Let us take the example directly. See, this is a alpha amino acid. Why this is alpha amino acid? Because at the alpha position we have amino group, right?
Yes,
This is
sir.
a form of alpha amino acid. This is again a alpha amino acid or not? They both are different amino acids. They are both different alpha amino acids, but both are alpha amino acids, right? This one is
Yes,
called
sir.
alanine and this one is called glycine. This is glycine, this is alanine. Alanine and glycine have one thing different. Here we have CH3 group, here we have no CH3 group, right? So what is happening? The OH part of the carboxylic acid of this glycine is reacting with the H of the nitrogen of this alanine, and they both are forming a bond by eliminating what? They are eliminating what? OH plus H is
Uh,
forming what?
uh, Sir,
If
bond
we,
with sir, OH will form bond and H will also form bond.
no. See, you have this OH and this H. You are eliminating them. Suppose you are removing them by connecting with each other and you are joining this bond directly here. What you are
Yes,
doing,
sir.
you are removing the water molecule and connecting these two sides.
Yes, sir.
So this H2O will be removed and the removal of H2O is called condensation and this bond will be formed. Here what we are seeing, this is directly connected with nitrogen here, right? And this bond is called what? Peptide bond. And this compound is called glyc what is it? Glycyalanine. We connected glycine with alanine. The compound name is this. And they might ask you which type of peptide bond is this. So this is dipeptide bond. You can understand it simply. If we have two amino acid unit, it will be dipeptide. If we are connecting three amino acid unit, it will be tripeptide. For four, tetrapeptide, right?
Yes, sir.
Carboxy group of glycine combined with amino group of alanine, we get dipeptide glycylalanine. This is important again. A dipeptide bond is formed by two amino acids. A dipeptide bond is formed by two amino acids. A tripeptide bond will be formed by three amino acids, and so on. More than 10, we will call it polypeptide. A polypeptide with more than 100 amino acid residues having molecular formula this is called a protein. Very simple definition. However, the distinction between polypeptide and protein is not very sharp. Polypeptide with fewer amino acid like protein is ordinary. Okay, that's easily nice. It contains 51 amino acids. This is just normal theory. The distinction between polypeptide and protein is not very sharp. Polypeptide with fewer amino acids are likely to be called proteins if they ordinarily have well-defined conformation. You can like they won't ask you a question from this. This is for just general knowledge, right? You can give it a read. The important thing is this. This bonding part you should be very clear with. What is happening? Two amino acids are coming to each other, coming closer to each other, right? The OH part of carboxylic acid will be reacting with H part of nitrogen and they will be removed in the form of H2O and the bonds will be connected simultaneous, right?
Yes,
And this
sir.
is called polypeptide bond. And if we are using two amino acid, we will call it dipeptide. And if three, then tripeptide, and so on. Now, classification based on molecular structure. Classification of proteins based on molecular structure. This is very, very important. They will directly ask you what are fibrous protein and what are globular proteins, right? So basically, they are proteins and they are classified or differentiated on the basis of molecular structure. Let us talk about what are these. A globular protein look like this, a spherical shape, right? Like we are bending the chain, it will look like this. A globular protein are spherical and function in transport of enzyme and regulation. A fibrous protein basically look like a cylindrical or straight chain type of thing. Fibrous proteins are elongated structure, providing support and strength to the tissue. So this is how they look like. And let us talk about what they have to offer. So first of all, fibrous protein. When the polypeptide chains run parallel, we have polypeptide chain. What are polypeptide chains? First of all, if we have connected more than 10 amino acids with each other, here we connected only two, right? If I
Sir.
have connected 10 or more amino acids in a similar way, I am just bringing the amino acids closer to each other and I'm eliminating the H2O, again H2O, again H2O is removed. And I have made a chain of 10 amino acids or more. Then I will call it polypeptide chain. Now, when polypeptide chains run parallel and held together by hydrogen and sulfide bonds, they then fiber-like structure form. And this is fibrous protein basically. What do we mean by polypeptide chain run parallel? Suppose this is my long chain consisting of 10 or more amino acids and they are running parallel to each other in a straight line, in a straight manner. They are running parallel to each other. If this is happening and these chains, suppose I have this polypeptide chain of amino acid, I have this polypeptide chain of amino acid, and I have this polypeptide chain of amino acids. If they are running parallel to each other and all of them have this type of bonding, hydrogen and sulfide type of bonding, suppose these two are attracted towards each other with hydrogen and sulfide bonds. These two polypeptide chains are forming hydrogen and polypeptide bonds with each other. Then this will call fibrous protein. Is it making sense? Or should I bring some
Sir.
GIF or animation type of thing?
Sir, it's making sense.
Okay, what else I can use? Mm, okay, okay. Suppose, see, do you have two pens?
Yes, sir.
Show me your two pens. They are parallel, like you have just merged them, like they are not straight, they are thread-like thing, thread-like. They are curving with each other, right? If they have hydrogen and sulfide bond with each other, if these two polypeptide chain have hydrogen and sulfide bonding with each other and they are running parallel to each other, then this will be fibrous protein and it will look like this. Is it making sense?
Yes, sir.
What else do we have? They are insoluble in water. They are chief structural material for animal tissue. They are chief structural material of animal tissue. The example of fibrous protein is keratin, present in hair, wool, silk, and myosin, present in muscles. So you might have heard shampoos with having keratin, right? Like
Yes,
this
sir.
shampoo contain keratin. So why keratin is important? Because it is helpful for the growth of hairs. So it is basically a type of protein, fibrous protein, and they are present in our hair as well, wool and silk, right? Now,
Sir.
globular protein. The structure results when the chain of polypeptide coil around to give a spherical shape. Now, in the case of fibrous, they were running parallel to each other. They were not bending. They were just running parallel to each other. If if this chain is not running parallel, they are just bending. Suppose I have a thread, I am just making a coil of it in a spherical shape. I am just making a coil, basically spherical. I am just circulating it. Then it will look like this, and it is type of globular protein. Right? Suppose, see, in the case of fibrous, we were having this chain. Like they were straight chains, but running parallel, having hydrogen sulfide bonding with each polypeptide chain. Like they were having hydrogen sulfide bonding. But if we are talking about globular protein, they are making a coil, like they are making a random structure like this. So if they are making a coil type of structure, spherical shape, they will be called what? Globular protein. Globular protein are soluble in water. Fibrous was non-soluble. These are soluble. The intermolecular forces are very weak. In case of fibrous, they had hydrogen and sulfide bonds, and hydrogen and sulfide bonds are relatively stronger bonding. But here the intermolecular forces are weak. The example of globular proteins are insulin and albumin. Do you know what is albumin and insulin?
There are no insulin, but albumin and
Okay, let me show you what is. Mm, Albumin is basically like I'm not able to show you a GIF sort of thing, but it is present in blood plasma and it is synthesized in liver, right? It is basically a type of protein which is inside our body, where in blood plasma, right?
yes,
If
sir.
you need to look at the GIF, they are not showing any GIF type of thing. Now, again, the differentiation, if they ask you the difference between them, you can say these are linear condensation product. They are running linearly, right? They are not making any coil type of thing. They are not bending. They are just running parallel with each other. They are cross-link condensation, right? Polymeric acidic acids and amino acids. They are insoluble and they are soluble in water. Polymers of acidic and basic amino acids. They are linear proteins. They are folded and give rise to 3D spherical shape. They have hydrogen bonding. They have weak intermolecular attractions. So this is just the difference and basically what we already talked about.
Yes, sir.
Now, we talked about 20 different type of amino acids joined together and form a protein. So these are the different type of amino acids and they join in this manner, right? Suppose if I have replaced, if I have replaced this glycine from first position to this, suppose I have replaced these two species. In place of glycine, I have placed this SER, right? Then I will generate an entire new type of protein. On the basis of sequence, we can generate different and different type of proteins, right? But the
Yes, sir.
basic condition is we need 20 type of amino acids. These amino acids basically. Now, this we already discussed. Neutral, equal number of amino group and carboxyl group. Basic, more number of amino group. Acid, more number of carboxyl.
Yes, sir.
We talked about what? Classification of classification of protein on the basis of molecular structure. Now, We will talk about the structure of protein itself. We have basically four type of structure: primary, secondary, then tertiary, and then quaternary. Right? We'll go into them one
Then,
by one. You have your class now. We'll
then
start in.
yes, sir. We'll start in
Okay.
the next class.
Listen. Do you have any doubts from the previous part? Like any doubts with how we are forming the structure of glucose, how it was linear, how we converted into chain, what was the reason? Have you gone through it or not? Have you revised this chapter?
Yes, sir.
Sure.
Yes, sir.
Okay, after this chapter, what we need to start? Organic, right?
No, sir. Haloalkenes and haloarenes.
Organic, basically. We will start the next chapter and we will try to complete this by tomorrow. So please
Sir.
try to study before coming. Okay. Thank you. That's it for today. Good luck. Bye bye.
Thank you, sir. Sir, my exams are also coming next month.
Next month,
June.
don't you have like
Yes,
summer
sir.
break?
Sir, after my exams, there are summer break.
Like your class is going to be till which date? From when your summer
Sir,
break will start?
sir, on July, but till June ending, all my exams will be over.
So your summer break will start in July, right?
Yes, sir.
July, complete
July,
1 month. Uh,
August, and it will reopen on September.
July, August, you will have your summer break. In June, you
Yes,
have
sir.
your another exam.
Yes, sir.
Do you know the
Midterm.
syllabus? Midterm
Yes,
syllabus?
sir.